So far in activity 3 and 4, you have looked at various types of cells, both plant cells, animal cells, protists, and some bacteria. And we need to take some time to talk a little bit about organelles and their functions, which you looked at in activity 4. So we will focus this particular discussion on cell organelles. Just a quick recap. We've talked multiple times about the two major types of cells, prokaryotes and eukaryotes. If you remember, prokaryotes, this is an example of a prokaryote in the picture below. Those mainly are bacteria. You can see in the center there is a section of DNA, and if you look very closely, there is no nuclear membrane around this. So the DNA is free floating in the nucleus or in the cytoplasm. You also notice that there are a few organelles, ribosomes, cytoplasm. There's a cell membrane and a cell wall as well. So again, to recap, these are all things that you should know. And if you don't, I would suggest that you write them down. These tend to be smaller in size than their counterparts, eukaryotes. They have simpler structures, and by that I mean they lack most of the organelles that you're going to see or have seen in eukaryotes. The DNA for these cells do not have a membrane, so they don't have a nucleus per se, but they do have genetic material. Eukaryotes, on the other hand, and again, this is an example of a eukaryote, and if you just take a quick glance and compare, you will notice that it is significantly more complex. You'll see many of the cell parts that you have just, or the cell organelles that you've just looked at, but most importantly, right in the middle of this purple structure, you will see a nucleus with a nucleolus. The purple outside part is a nuclear membrane, and the little dots that you see are nuclear pores, which allow stuff to come and go from the nucleus. So these types of cells are larger than the prokaryotes. As you can see just by a glance, they are much more complex in structure. And as I mentioned before, they have that nucleus in the middle that contains their DNA. And again, all of this should be review. However, if there's one bullet point or whatever that you don't remember, you might want to write it down, or perhaps even use a Venn diagram to organize this particular set of information, because this is a very good compare-contrast type scenario. So just recapping quickly, cells, two types. And again, I'm going to abbreviate these for the sake of simplicity, but they can be broken into prokaryotes, which lack a nucleus. And again, if you think graphically, this may be a way for you to represent some of your thoughts in your notebook if you wish. So prokaryotes don't have a nucleus. Eukaryotes, and again, I'm abbreviating this, do have a nucleus. Prokaryotes are mostly bacteria. And this can be bacteria or it can be archaeobacteria. We've spent a little bit of time talking about archaeobacteria last week. The eukaryotes, there are four major groups. You've looked at plants under the microscope. You've looked at animal cells, and again, abbreviations under the microscope. Fungi you have not looked at under the microscope. But again, if you go back to activity 2.5, you will see many of the characteristics of a fungus. And last but not least, you have looked at protists. Whichever one you looked at, whether it was an amoeba or the paramecium, both of those are examples of protists. So the plant that you looked at in activity 3 was Elodea. The cheek cells were animal cells. You didn't look at this. And then this was the amoeba or the protist. The bacteria you looked at were either circular or spiral in shape. In talking about eukaryotic cells, there are two major types that we are going to focus on: plant cells and animal cells. And again, this was the focus of activity 4. So most of this should be a review for you. As you look through these next several slides, really what you should probably be doing is thinking about what you need to add versus writing everything down. Both plant and animal cells have many different organelles, and this has not really been defined to this point, but an organelle is just a structure that carries out a certain function. For example, ribosomes make proteins. The nucleus controls all the activities within the cell. So all of the things that you drew within your cell for activity 4 would be examples of organelles. And as you can see, there's a whole host of organelles. And by all means, this is not a complete list. These are the major organelles that we are going to focus on. And not only thinking about what each of these does, but you should also think about how they interact with each other to complete a job. For instance, you should be able to look at this list and say, okay, so here's who's involved in making a protein, and here's what they do to contribute to making a protein. So don't just look at this list and say, okay, I just need to know what they do, but you need to think about how they interact with each other to finish a common product for the cell. We're going to now look at each of the individual organelles. The nucleus, and that is this guy right in the middle that I'm outlining. The outer part of the nucleus is called a nuclear membrane. And again, you may want to highlight this and label the nuclear membrane just so that, again, it's a part that you should know, and it contains nuclear pores, and it is the part, just like the cell membrane, that holds the nucleus together. The nucleus is the control center of the cell. It contains DNA and eukaryotes, and it also directs protein synthesis. You have heard about proteins before. At the beginning of the year, we talked about the building blocks. Amino acids put together make up proteins. And this is a word you should also be familiar with because we talked about dehydration synthesis. So in that instance, synthesis was the making of water. And here, this is going to be the making of proteins. So the nucleus is directly responsible for helping make proteins in the cell. The nucleolus is this darker center in the middle. And again, it sits in the inside most of the time. It kind of looks like an egg yolk of a nucleus, and it makes ribosomes. So just this part is responsible for making ribosomes, and those ribosomes are used in making proteins. So those ribosomes are transported out of the cell into the cytoplasm so that they can function in making proteins. When we talk about, again, this nuclear membrane has to be somewhat permeable so that material can come and go. The DNA that's in it must be accessible at all times, and we also need to be transporting ribosomes out of the cell. So again, this little membrane has pores in it, which allow things to come and go, to access the DNA, and be able to export ribosomes. In the nucleus, you'll also find the chromatin. We will talk more about chromatin later when we talk about cell division, so don't get too hung up on exactly what this is, but it's a stage of DNA that we will, again, talk about in the future. We've already talked about ribosomes significantly. You find ribosomes in 2 locations in the cell. You'll find them free-floating in the cytoplasm, and then you'll also find them attached to the endoplasmic reticulum. If the endoplasmic reticulum has ribosomes on it, it is called rough endoplasmic reticulum. And the complementary match to this would be smooth endoplasmic reticulum. They do have different jobs, and we'll talk about that in just a second. But I just wanted to point out that there are two different types of endoplasmic reticulum. If it has ribosomes on it, then it's considered to be rough. Ribosomes are involved in producing proteins. And again, if you didn't write it down before, synthesis is the word that we use when we talk about making proteins. So this process is called protein synthesis. And again, we will talk about that after winter break in more detail. It contains, these guys deal with instructions from the nucleus. The nucleus has information on how to make proteins. And again, these are either free-floating or they're attached to the rough endoplasmic reticulum. So once again, as we move through these cell organelles, think about how it best suits you to add the information you already have or to organize this information. Maybe it's a chart, maybe it's some type of diagram, so that you can easily access what these particular organelles do and how they interact with each other. All right, we've already mentioned endoplasmic reticulum. This is a set of membranes that connect the nuclear membrane to the cell membrane. And for the most part, because they're kind of like a highway, their job is to transport. If you think about the endoplasmic reticulum in terms of everyday things that you might see, this would be like 270 that goes all the way around Columbus. It's involved in transport of people. So this particular set of membranes is involved in transport from the nuclear membrane anywhere to the cell membrane. So again, it's kind of like 270 or any highway that you might be familiar with. I already mentioned that there's two types. This rough endoplasmic reticulum has ribosomes, and the smooth endoplasmic reticulum does not. Please note, and we'll look at this in the next two slides, that these do have different jobs. In addition, if you're not really fond of writing out endoplasmic reticulum, you can abbreviate rough endoplasmic reticulum as RER and smooth endoplasmic reticulum as SER. It just saves you from having to write this out every time. If you use RER or SER, any biologist will know what you're talking about. So don't think that these are universal terms. So let's talk a little bit more in detail about the rough endoplasmic reticulum. You can see this is a really nice diagram that shows the rough endoplasmic reticulum has the ribosomes on it. They make proteins for the membranes. And the smooth endoplasmic reticulum does not have ribosomes. And what they do, they have enzymes. Enzymes help move along chemical reactions so that you can make, again, here's this word, synthesize. Synthesize means to make. They help make certain molecules like lipids. And these lipids are used for a variety of purposes within the cell. Again, the enzymes are things that help move along chemical reactions or help speed them up. And synthesize means to make. Okay. They also, the smooth endoplasmic reticulum also helps in breaking down drugs and alcohol that the cell may encounter. So they're kind of a detoxification organelle as well. If you'll notice, this set of membranes, one side of this would connect to the nucleus, and the other side would connect to the cell membrane. In many diagrams that you see, however, this won't necessarily stretch all the way from the nuclear membrane to the cell membrane. So don't be misled if you see endoplasmic reticulum that doesn't touch the cell membrane. The Golgi apparatus, if you look at it and use your imagination a little bit, it looks like a stack of pancakes, and it looks like it has maple syrup dripping off the edge of the pancakes. So if that helps you to remember what this looks like, fine, and if it doesn't, that's fine as well. You can see that this is a flattened structure, and you can even see some of the little vesicles in this example that are on the edge of the Golgi apparatus. Basically, this particular organelle serves in packaging and modifying and secreting different cell products. For the most part, it specializes in ribosomes. So it will, inside of the structure, it will modify a protein, and then it will use a vesicle to transport that to where it needs to go in the cell. So this is a good example of how one particular organelle modifies proteins or packages them, puts a bow on them so they can be shipped out, and then the vesicle does the transport for that particular organelle. So, one organelle works with another organelle to be able to transport proteins. Lysosomes contain, here's another, the word enzyme again, contains digestive enzymes. So basically what these do is speed up the digestion of food or organelles or any other waste that comes into the cell. If you'll notice that in this instance, is a really nice diagram. Food comes into the cell, a vacuole surrounds it, and if it weren't for the lysosomes, notice the lysosome attaches to it, and you can see by the white stuff that's being excreted into the cell, those are the digestive enzymes which help break down the material. This vacuole then, or this lysosome, then detaches and will wait for the next food vacuole or whatever it needs to break down. But the remains of this are then exported out of the cell and eliminated. So again, you can see a Golgi apparatus. You can see that there are lysosomes that are being, packaged and secreted here. These lysosomes work with food vacuoles that have materials to break it down, and then it's exported via that vacuole. So again, as you look at these diagrams and you think about what cell parts do or cell organelles do, ask yourself what other organelles are involved in that organelle's job, because there aren't very many of these organelles that work all by themselves. They all have partners in crime. Lysosomes, you will find numerous lysosomes in animal cells. However, it's still debatable whether or not plant cells have lysosomes. So, many scientists believe that they're there, others don't. So normally if you see lysosomes, it's an indicator that it's a plant cell or an animal cell, because you'll see quite a few of them. We just talked about vacuoles in the previous slide. Vacuoles are huge in plants. If you look, this is a plant cell, and almost half of the cell is made up of the central vacuole. Mainly what they do are store nutrients or water or waste products. Particularly in plants, they store water. So if you see a plant that's shriveled up, that means all of the water has left its central vacuole, and the cell does not have any rigidity anymore. If you look here again, this is your central vacuole. In animal cells, these are much tinier. All right, so you can see there's several little vacuoles in this animal cell. When we talk about vesicles, again, vesicles are involved with other organelles in transport. Most of the time, they're small membrane-bound sacs that divide the materials that have come into the cell from the rest of the cytoplasm. Their main job is transport. So if you'll notice, this guy is bringing in food. The cell membrane pinches off. This is going to be your vesicle. The vesicle is then going to, in this instance, attach to a lysosome. It's going to digest the food, and then this is going to continue to be transported out of the cell, if that be the case. Most of these are pretty short-lived, but they form and then they're recycled as needed. Most of you mentioned mitochondria when we started the Who's Who activity as something that you remembered. This is an artist's image of a mitochondria, and then this is an actual electron microscope image. You can see all these little folds that are in it, are these little finger-like projections. Probably the most important thing to see here is if you notice there's an outer membrane and an inner membrane to the mitochondria. And from the endosymbiotic theory video, if you remember, this is evidence to support that once upon a time this was a bacteria, and this outer membrane is probably the membrane of the organism that engulfed it. These guys are considered to be the powerhouse of the cell because they make cell energy. We will talk about ATP when we talk about cellular respiration, but for right now, what you need to know is that ATP is cell energy. While you may eat a candy bar and that glucose is taken into your bloodstream, that glucose has to be turned into cell energy. It's not, the glucose itself isn't super usable by the cell. So that has to be turned into something the cell can use, and that's called ATP. These little finger-like projections that you see are called cristae, or here you can see it's crista because it's a singular, and these folds are super important. Again, we'll talk about this later, but all of these folds increase surface area. And for the mitochondria, more surface area means more ATP production. Also, if you remember, just a side note, these guys have their own DNA and their own ribosomes, which again, a piece of evidence for the endosymbiotic theory that these were probably their own bacteria at one point. Chloroplasts are unique to plant cells only. Most of you, again, remembered that from middle school. They contain a pigment that's called chlorophyll, which gives them their green color. All of these little tiny dots that you see are chloroplasts. The outer edge that I'm outlining right now is the outside edge of a cell. So you can see there are multiple cells within this slide. And you should go back and look at activity 3 in your drawing of the Elodea leaf. Your drawing should be very similar to this. So you should see these long rectangular blocks, and you should see these little dots, which are the chlorophyll that gives them their green color. The chlorophyll and the chloroplast capture sunlight, and it helps produce food for the plant through photosynthesis. And again, we will talk more about photosynthesis in the weeks to come. But again, remembering back to endosymbiotic theory, these guys have their own DNA, they have their own ribosomes, so we are looking at something that was probably a bacteria that was living on its own before it was engulfed by a larger bacteria. Again, in the future, we're going to talk about the cell membrane in more detail. This is a great image of a cell membrane. Actually, our next unit deals with the cell membrane and cell transport, and we're going to talk more about what all of these parts do. But for right now, what these do is control what comes and goes from the cell. Just like the nuclear membrane, things have to come and go. The cell membrane does the exact same job. They provide protection and support. They're basically made up of lipids. And if you remember, we talked about lipids when we talked about the endoplasmic reticulum, and they also have proteins in them. When we head into our next unit about cell transport, we're going to talk a little bit more about these proteins and what they do. The cell wall is an additional layer to plant cells for the most part. I know the slide says plant cells only. If you go back and take a look at your Who's Who activity, you'll remember that bacteria and fungi also have cell walls. But again, for the most part, when looking at plant cells, we consider the cell wall to be a distinguishing organelle for it. When you look at the cell wall, it's always found on the outside of the cell membrane. So if the cell membrane is this inner part, then the cell wall is the part on the outside of it. You can see, I just want to point out, again, here's a huge vacuole. Here's your chloroplasts. Notice that plant cells also have mitochondria. And then here's a Golgi apparatus and some rough endoplasmic reticulum in this cell. your nucleus, your nucleolus as well. The cell wall is very rigid. However, it is also permeable. So what that means is it's going to allow stuff to come and go from it. It's not so rigid that it won't let anything pass. Otherwise, you wouldn't be able to get nutrients in and out of the cell. Cytoplasm, we've talked about this multiple times. Actually, I'm going to go back one slide and point out that cytoplasm is everything, for the most part, that's not an organelle inside of a cell. So from nucleus to cell membrane, that's all going to be cytoplasm. Cytoplasm is kind of jelly-like. So if you think of clear jello, it is the organelle that holds all the, it's the organelle that holds all of the other organelles. It's kind of like a shock absorber. So it protects those organelles if something would happen to the cell, like if it were jarred around, it provides a little bit of a cushion. It's also really important because most of the cellular activity happens here. There are lots and lots of chemical reactions that happen within the cytoplasm. Just like most of the organelles, we will talk more about centrioles and centrosomes when we get to cell division. These are found exclusively in animal cells. And a centriole, and this is a centriole, and this is a separate centriole. So you may want to go back and look at your drawing or maybe draw this at this point so that you can tell the difference between these two. So this set of microtubules is a centriole. This is a centriole, but the whole structure, two centrioles, make a centrosome. Right, so again, however it best works for you, I would think about drawing this or making sure that you understand that centriole, centriole, centrosome. Okay. These guys are involved in spindle production during mitosis, and try to jot down a note to yourself if you can remember what mitosis is. I know we've talked about it in the last couple of weeks a little bit, so I know that you remember. But go ahead and jot that down, and then we can talk about that in class. But these are involved in making spindles that are used during mitosis. So now that we've talked about different organelles for both plants and animal cells, it would probably be beneficial for you to think about things that just the animal cell has, just the plant cell has, and then things they have in common. So you probably want to spend a little time to go back and look at those things and make sure that you are clear on what makes an animal cell unique, what makes a plant cell unique, and then what things do they have in common. Feel free to go back and look at any of the organelles in more detail. While you are listening to this, I will be around to be able to help and answer questions as well.